Caloric density versus nutrient density. Most gym-goers track total daily calories without examining the underlying metabolic matrix of their food. 1,000 calories derived from refined carbohydrates triggers an immediate insulin surge and rapid gastric emptying. The exact same 1,000 calories from whole animal proteins and healthy lipids requires hours of complex enzymatic breakdown. Your cellular machinery does not register all caloric units equally. Nutrient density dictates hormonal signaling pathways, satiety mechanisms, and micronutrient absorption rates. Consuming high caloric density devoid of essential micronutrients leaves muscle cells in a state of functional starvation. Even in an energetic surplus, deficient nutrient density leads to visceral fat accumulation while halting lean muscular hypertrophy. Evaluating your diet strictly by numbers on a scale while ignoring micronutrient density sabotages long-term physical adaptation. Here is what happens under the surface when nutrient density is ignored. Systemic inflammation rises, insulin sensitivity degrades, and cellular repair processes grind to a halt. To optimize body composition and physical output, energy intake must be matched with high biological value and dense cellular nutrition. The protein leucine threshold. Consuming protein continuously throughout the day is ineffective if individual meals fail to hit the leucine threshold. Leucine is the principal branched-chain amino acid responsible for activating the mTORC1 signaling pathway. Without approximately 2.5 to 3 grams of leucine in a single bolus, muscle protein synthesis is barely triggered. Spreading 20 grams of total protein across 6 micro-meals keeps circulating leucine levels below the activation trigger. 3 to 4 substantial protein feedings containing rich leucine sources yield significantly higher net anabolic signaling. Total daily protein intake provides the raw building blocks, but single-meal amino acid thresholds dictate actual tissue repair. If your meals do not cross this biochemical threshold, your body remains in a neutral or catabolic state despite eating protein.
What most lifters fail to realize is that meal frequency without leucine saturation is metabolically wasteful. Ensuring every major meal contains sufficient leucine is essential for maximizing muscle protein accretion over time. This simple adjustment in protein distribution often restores muscular growth in stagnant lifters.
Carbohydrate glycogen resynthesis. Carbohydrates are frequently mischaracterized as a fat storage driver rather than an essential performance substrate. Intramuscular glycogen is the primary fuel source for high-intensity muscular contractions under heavy resistance. When muscle glycogen stores are depleted, force output drops sharply and muscular breakdown accelerates. Carbohydrates consumed post-workout are preferentially partitioned directly into muscle cells via GLUT4 glucose transporters. This non-insulin-dependent pathway rapidly replenishes glycogen without spilling over into adipose tissue storage. Restricting carbohydrates during periods of intense hypertrophy training compromises mechanical tension and muscular fullness. Carbohydrates do not cause fat gain under a controlled energy balance. They keep muscle cells fully hydrated, preserve cellular volume, and drive maximum force production in the gym. Here's the biological reality of low-carbohydrate training.
Without sufficient glycogen, thyroid hormone conversion drops and cortisol levels rise during hard sessions. Proper carbohydrate timing shields muscle tissue while driving optimal glycogen resynthesis. Micronutrient bioavailability. The total quantity of a vitamin or mineral listed on a food label is rarely the amount your body actually absorbs. Plant-based micronutrients are frequently bound to anti-nutrients such as phytates and oxalates that inhibit intestinal absorption. Heme iron sourced from red meat possesses a 3-fold higher absorption efficiency than non-heme iron found in spinach. Zinc, magnesium, and fat-soluble vitamins require specific digestive cofactors and healthy lipids for cellular uptake. Evaluating your daily nutrition purely by raw label numbers leads to widespread subclinical micronutrient deficiencies. Subclinical deficiencies impair enzymatic reactions, energy production, and hormonal recovery. Bioavailability determines what actually reaches your target tissues, not the theoretical values printed on the package. This is where the biology becomes critical for serious lifters.
A diet high in unrefined anti-nutrients can bind key minerals before they cross the gut barrier. Prioritizing highly bioavailable food matrices ensures your physiological engine receives usable micronutrients.
The thermic effect of food. Not all macronutrients require the same energy expenditure during digestion, absorption, and metabolic conversion. Dietary protein carries a thermic effect of roughly 20 to 30%. This means nearly 1/3 of the total calories consumed from protein are burned simply processing the amino acid chains. Carbohydrates require 5 to 10% of their energy for digestion, while dietary fats require less than 3%. A high-protein diet intrinsically elevates your daily basal metabolic rate compared to an equal-calorie high-fat diet. Ignoring the thermic cost of individual macronutrients creates major inaccuracies in energy balance calculations. Prioritizing protein creates a metabolic advantage that supports fat loss while preserving lean structural tissue. What happens next in energy partitioning is remarkable. The metabolic energy required to convert amino acids into glucose via gluconeogenesis further increases total calorie expenditure. Protein dominates metabolic efficiency through both thermic cost and tissue preservation.
Lipid membrane permeability. Dietary fat is not merely an energy storage medium, it is the structural backbone of every cell membrane in your body. Essential fatty acids, particularly omega-3 and omega-6, govern cell membrane fluidity and insulin receptor sensitivity. Rigid cell membranes caused by excessive trans fats and processed seed oils impede nutrient transport into muscle cells. Cholesterol is the essential steroid precursor for endogenous testosterone, cortisol, and vitamin D synthesis. Cutting dietary fat too low collapses systemic hormone production and degrades joint synovial fluid. Consuming high-quality saturated and unsaturated fats maintains structural cellular integrity and optimal endocrine output. Here is what most popular diet advice fails to explain. When cell membranes lack omega-3 fatty acids, insulin resistance increases and muscle nutrient uptake slows down.
Balancing lipid intake keeps your endocrine system robust and your muscle membranes highly permeable to growth signals.
Circadian nutrient partitioning. The human body does not process nutrient intake with identical metabolic efficiency at all hours of the day. Insulin sensitivity in skeletal muscle tissue peaks in the early hours of the day and immediately following heavy resistance training. Consuming large carbohydrate-heavy meals late at night occurs when muscle insulin sensitivity is diminished and movement is minimal. Aligning your largest carbohydrate feedings around your workout window optimizes glucose storage inside muscle glycogen stores. Nutrient timing does not replace total caloric balance, but it dramatically dictates whether calories are partitioned toward muscle or fat. What happens when timing is ignored is subtle but destructive. Chronic late-night heavy carb loading elevates resting blood glucose and inhibits nocturnal growth hormone release. Matching carbohydrate intake to metabolic demand keeps insulin working for your muscles rather than your fat stores.
Net nitrogen balance. Structural muscle growth cannot occur unless your biological system maintains a positive net nitrogen balance over time. Nitrogen is a fundamental chemical element contained exclusively within amino acids and structural muscle protein. When nitrogen excretion through sweat, urine, and metabolic waste exceeds intake, your body enters a net catabolic state. Heavy resistance training creates an immediate negative nitrogen balance by inducing microtears in muscle fibers. Sufficient high-quality protein consumption paired with adequate recovery shifts the nitrogen balance back into positive territory. Hypertrophy is ultimately the mathematical result of keeping total nitrogen accretion higher than daily nitrogen loss. This is the ultimate rule of human muscular biology. Without a constant positive nitrogen balance, no amount of heavy training or supplement use can force muscle growth. Maintain consistent protein delivery, protect your recovery windows, and let biological physics handle the rest. The human body is not a simple calculator of calories in and calories out. It is a complex biological engine governed by hormonal signals, enzymatic thresholds, and tissue bioavailability. Master the fundamental biology of your nutrition, and your physical performance will follow.